What are the functions of the complement protein? MAC formation, all agree with this? Okay, what is the other function? What does C3A, C4A, C5A can do? Anaphylotoxins, okay? And what was involved in anaphylotoxin? Smooth muscle constriction, vascular dilatation, increased permeability, and calling other immune cells. Then what is the other function? Opsonization. What does opsonization mean? Tag the bacteria so that it can be eaten up by the macrophage. Okay, so things are not as bad as it looks. Next thing is inflammatory reaction. So same chemotaxis, these complement proteins, when they are in the blood, they can call other immune cells. Neutrophils, please come to the site. Macrophages, please come to the site. Here there is a fight going on, because you should think of everything, whether it is an autoimmune disease, a pathogen in your body, a bacteria, a virus, endotoxin, a fungi. Complements, whenever are activated, they will call other immune cells toward the site of inflammation. Now this is a slightly different function which you would expect. Complement proteins not only help in infection, but it also helps in maintaining your normal physiology or normal functioning of the body. How it does it? So a lot of immune complex will be formed. So whenever a pathogen comes, antibodies will be produced against it, and antigen antibody complexes will be formed. And later on, when the job is done, they need to be cleared from the system. You cannot leave them like that, because those complexes can deposit in the kidney, can cause glomerulonephritis. They can deposit in the lungs, at the alveolar membrane. So they can cause damage to your body, because they are, whenever antibody is alone, antigen is alone, it is soluble. But whenever they bind to each other, then they form complexes or precipitate. These precipitates can damage your kidneys, your lungs, and vessels, cause vasculitis. So they need to be removed from the system. Okay, this clearance is also done by the complement proteins, the different complement products that we saw. For example, C3b in particular, what it will do. So for example, a lot of antibodies have made a big network of immune complexes, like the one you see here. The red blood cells are in the figure with lot of antibodies around it. They can make them small, so they will detach them. So whenever C3b is produced, it will first break it up into small complexes so that they are not big in size and do not deposit. That's number one. Number two, red blood cells have got a complement receptor that will bind to these immune complexes, and then it will take it to the spleen or a lymph node, like a truck, like any vehicle, to transport them to the spleen so that they can be eaten up by the macrophages in spleen and liver. So clearance of the immune complexes is a very important function of the complement proteins. So the new functions that we talked about: opsonization means tagging of the bacteria. Number two, we talked about the chemotaxis, they will call the immune cells. Number three is when immune complexes are formed, they need to be broken up into small pieces and then removed from the system. Now we'll see the clinical consequence of this. There are two types of hemolysis that take place: intravascular hemolysis and extravascular hemolysis. Intravascular hemolysis, one of the important example that we know is Abo Incompatibility. Okay, so whenever by mistake or for some reason you gave blood group B to someone who has got blood group B positive, or for example you gave blood group B to someone who has blood group A, that is, there is Abo incompatibility. So what happens immediately? There is a transfusion reaction and there is a lot of hemolysis. Why that take place? Because everyone has, so if I am blood group A, which preformed antibodies I will have? Anti-B. Anti-B are going to immediately attack the red blood cell. So this first example you can see here is antibody, and they are in which type of immunoglobulin? NTB. IgM or IgG? IgM. Do you all agree that it's not IgG? And what could be the reason for it? And you have also studied the RH incompatibility? Yes. RH incompatibility occurs because of which immunoglobulin? IgG. Why not IgM? No, my question is that red blood cells have got whatever antigen, okay? NTA, NTB or RH antigen. In RH incompatibility there is IgG. In Abo incompatibility there is IgM. Why? There must be some reason. So there is a small point you need to remember, that our B cells, they can make IgG and IgM. IgG are mostly produced against protein antigens, and IgM are mostly produced against carbohydrate antigen. Abo antigen is carbohydrate in nature. RH antigen is protein in nature. So in RH antigen, when IgG antibodies are produced, they can cross the placenta and cause RH incompatibility. While Abo antigen, they will only produce IgM against them, and IgM cannot cross the placenta. So here, in the Abo incompatibility, IgM is important. In RH incompatibility, IgG is important because of the antigen that is causing its production. Do you get this point? Yes. So IgM is produced against the Abo antigen. What will happen? Which pathway will be activated? Classical pathway. IgM will activate the classical pathway C1, and MAC formation will take place inside the blood. As a result, what will happen? Red blood cells will be destroyed inside the vessel, intravascular hemolysis. What are they going to release? Hemoglobin. So that's why there is hemoglobinuria, and heptoglobin levels will come down, because free hemoglobin is bound to the heptoglobin. It will be used. So heptoglobin levels are down, hemoglobinuria is seen. Now on the contrary, when there is RH incompatibility, or whenever there is some other condition where IgG is produced, like autoimmune conditions, this IgG is going to bind to red blood cells, and this binding will mostly take place in the spleen. They will bind to the red blood cells. Now they will act as opsonin. Opsonization will take place. They will activate the macrophages. The macrophages will engulf it inside, and extravascular hemolysis take place. So another thing you need to remember is IgM, one molecule is enough to activate the classical pathway, but 200 molecules of IgG are required to activate the classical pathway. That's why mainly it's the IgM that activates the classical pathway, and intravascular hemolysis is dependent on IgM, and there is rupturing of the cells inside the blood vessel. While IgG, it will bind to the red blood cell, act as an opsonin, helps the macrophage to engulf it and destroy it in the spleen. And the example is autoimmune condition SLE, systemic lupus erythematosus. So these are the two examples, just for the sake of understanding that how the complement is involved, whether it is intravascular hemolysis, and how the complement is involved if there is extravascular hemolysis. Now we so far saw that complement proteins are activated and they will perform MAC formation, opsonization, phagocytosis, clearance of the immune complexes. At the same time, you need to keep a check and balance. If it keep happening, this MAC formation can attack any tissues. So you have to do a regulation for them as well. We will talk about only 3 of them. One is called as C1 inhibitor. So if you look at this figure, its name tells you that it's going to inhibit C1 of the which pathway? classical pathway. So here is, C1 has got CQ, CR and CS. C1 inhibitor simply remove R and S from the Q component, and C1 will not be able to function. So it's like there is a complex of three things: C1Q, C1R, C1S. They remove the R and S from C1Q and it won't function. It is C1 inhibitor. Number two is, you saw that whether it is classical pathway or alternate pathway, there is a C3 convertase step. C3 convertase of classical has got which components? C4b and C2a. Here C3 convertase has which components? C3b and BB. Decay acceleration factor is again going to remove these two components. So if you look at this figure, on one side C4b and 2a, which are C3 convertase, they are removed from each other, and on another hand C3b and capital B and small b, they are removed. So basically stopping C3 convertase. So when you stop them, remove them from each other, the pathway will not go further. Now we'll come to all the situations. So in the laboratory, you can measure the level of complement proteins. You can measure C3 level, C4 level, different, C3b, the active components, and then you have to find out whether the patient has got some condition or disease or not. So first thing, gram-negative septicemia. Do you all know what is gram-negative septicemia? No. Okay, so lots of gram-negative bacteria when enters your body and your body cannot clear it out. It spread in different organs, systemic spread of lot of bacterial infection, gram-negative bacterial infection at a very late stage, severe form of infection which can result in death. That kind of infection will have complement levels very reduced. And the reason is because antigen-antibody complexes will activate classical pathway, lot of them will use the C3, C4, C5, and usage will reduce the level of the complement. It is simply called consumption. So let's say you have 50 molecules of C1, 50 of C3. When lot of infection come, these 50 will be used. So after a while, when you check, the levels have dropped from 50 now to 10 or maybe even 5. The reason is because all of them is used. So whenever the levels of complement proteins are done, it will be found reduced in disease. Okay, that's very important, and the reason is because it is consumed, it is used up. Then in myocardial infarction, when there is tissue damage, classical pathway can also be activated by dead cells. So immediately after myocardial infarction, if you see the complement levels in the blood, it will be reduced again because that has been used. Autoimmune hemolytic anemias, so patients of SLE, rheumatoid arthritis or Sjogren's syndrome, autoimmune diseases, if you look at their complement levels, the complement levels are again reduced, and the reason is because it activates these pathways, they are used, consumed, and the level drops. And this particular slide just tells you that in the laboratory we can detect C1Q, C3, 4, 5, and the only condition that will elevate the complement protein level is during any inflammation, and the reason is not because it is helpful. The reason is that liver start producing acute phase reactants quickly during any kind of inflammation. So its increase is just as part of all the other proteins that are produced by the liver. Its more usage or importance is when its level are reduced and it is consumed because of its functions. Now let's see if you people can, if you focus here on these three pathways and then you look at these three different conditions that are mentioned here, we'll try to interpret them and see if you can understand it. So the first scenario is that C4 levels are reduced, okay, and C3 levels are normal. Now what does that mean? Which of these pathways is involved? First, so whenever you look at the complement protein levels, you have to decide that which pathway is involved. And then you can go to the beginning, whether it is immune complex region, whether it is bacterial polysaccharide, whether it is the mannose that is causing the activation. So when C4 is reduced but C3 levels are normal, which pathway you think is activated? Classical pathway. And what are the conditions? So the classical pathway is produced as a result of antigen-antibody complexes. So one condition is SLE, autoimmune condition, which has a lot of immune complex formation. Another is genetic deficiency. Why you think genetic deficiency? Sorry? Yeah, so genetic deficiency means that if somebody, a child has this defect, they will not be able to produce this protein normally. Its synthesis will not be normal. So once it's not normal, you will find in the lab the levels are very low. So one reason is genetic defect, one is antigen-antibody complexes are formed too much. We talk about SLE, and there is one more condition, hereditary angioedema. You saw I told you C1 inhibitor. What was the function of C1 inhibitor? It inhibits C1, separates Q from R and S. Some people have defect in this regulatory enzyme, so they don't have C1 inhibitor. So what will happen? C1 will be activated all the time, and it will keep producing MAC formation, anaphylatoxins, and these patients have got edema. Blood vessels are dilated and they have got edema around the lips and the face. So this condition, when too much use the classical pathway, that's why the C4 levels will reduce. And another important reason is because C3 levels are much more. In the body, we have got 1000 microgram of C3 and 600 micrograms of C4. So the levels are already half. And if they are used more, they will drop down very quickly. Now in the condition 2, if C4 is normal and C3 is dropped, what does that mean? Which pathway first? Alternative, because here C4 is not involved at all. It starts with C3, B and C5 convertase and so on and so forth. So if this is the reason, which could be the bacteria which activates the alternate pathway? The encapsulated bacteria like Haemophilus influenza, like Streptococcus pneumoniae, these have got encapsulated, these are encapsulated bacteria with polysaccharides. So if anyone has got gram-negative bacteria, then it will activate alternate pathway and C3 levels will be dropped while C4 will be normal. And if both levels are dropped, what does that mean? Both the pathways are activated. So you can see sepsis, heavy load of bacteria that includes encapsulated bacteria, that include antigen-antibody complex formation together. So when both pathways are activated, they will result in C3 and C4 levels will be dropped together. Any questions? I took my time to wait whether you will ask the questions or should I ask. Any questions? Nobody's asking any question. Sepsis is, at the moment, just remember, huge or massive bacterial infection. Okay? Or systemic, huge systemic bacterial infection is called as sepsis. You mean this interpretation or from the beginning? Okay. Let's make it simple. Let's just try to, use the information that you learned today. If you go to the lab, take a blood sample of someone who is suffering from sepsis, who has got gross bacterial infection. Now, Definitely, if somebody has got both gram-positive, gram-negative, large quantity of bacterial infection, of course, such a patient will be admitted in a ward. He will be in ICU or emergency. His blood will have antigen-antibody complexes. They will activate classical pathway. They will activate alternative pathway, and lot of complement will be used up. So if you measure his C3 and C4 level, we are just talking about two proteins that we will measure, C3 and C4, they will both be dropped. Does it make sense now? That because there is classical pathway activation, there is alternate pathway activation, they both have C4 and C3, all is used up, so the levels come down. And that is what you see in the report. Now, on the other hand, if there is certain condition that just activate the classical pathway, not the alternative pathway, and just one thing I forgot to write, if I write it, maybe it will make more sense. So if you look at the levels of the complement protein, C3 is around 1200 micrograms. C4 is only 600 micrograms. First, normally your body has got C4 half of that of C3. Okay? Now if you imagine classical pathway is activated very fast, it's using a lot of C3. Which one of these will finish first? C4. It's, common sense. That's why in the first group, where I said that the classical pathway, if activated, C4 is dropped, but C3 is still normal. Why? Because of this level. C3 is still more in quantity, C4 is less in quantity. So if the classical pathway alone is activated, it will first use C4, then the C3 levels will drop. So the first group, and the SLE, it is one of the autoimmune disease in which lot of antigen antibody complexes will be formed. We'll talk about this in one of the lectures in future. So in that condition, classical pathway is activated a lot of C4 is used, C3 is still normal. So first group is clear to everyone? Assalamu alaikum. And the second group in which C4 is normal, that is very clear, that only classical is not used, it's the alternative pathway.